Hi, this is a lecture for my Anatomy and Physiology 2 students, and this is the second part of eye and vision. All right, so the first thing we're going to be talking about for this lecture is light refraction. And refraction means the bending of light. It occurs when light passes through different media. So when we're talking about vision, we are talking about the air and also the structures of the eye. Now, light is refracted or bent 3 times before it hits the retina of the eye. And those 3 places are at the cornea, entering the lens, and exiting the lens. So as they move through those structures, that's going to cause a little bit of bending or refracting. Now, if you look at the top picture, it shows you a little, it shows you the pathway of the bending. And ideally, even though it gets bent 3 times, ideally we want that light to hit the retina. We want it to kind of dead end on the retina. And that's how we get our best vision. Now, because of this bending, if you look at that top picture, by the time that image gets to the retina, it will actually be upside down and reversed. The image is upside down and reversed from what the light rays are bouncing off of. Let's say it's a chair, or bouncing off of a chair. The light rays bounce off a chair and they enter into your eyes. They get bent those 3 places, and then by the time if we track it to the retina, it is that image would be upside down and backwards. Now, we don't see it as upside down and backwards, or I scratch that. We don't perceive it as upside down and backwards. That flipping actually occurs in your brain, which is pretty neat that your brain does that for you. Now, so before we move on from that, let's remember, because of light refraction, the goal is to get those light rays to converge on the retina itself. That's where the photoreceptors are. Now, one of the structures that light has to go through is your lens. And the lens is actually pliable. It is, it's actually movable or pliable. Now, your lens because it is pliable, we say it can accommodate. It can actually change shape. So how your lens accommodates is by changing shape. Now, if we look at the picture, the second picture down, you see kind of a thinned oval and then more of a sphere circle. Now, this represents what the lens can actually do. The lens can thin out or the lens can bulge. Now remember, the light rays are going through the lens and it's causing some bending or refracting. Now I do have a video, an animation for you to watch. We can't do it here. I can't click on it here because this is going to be made into a video. However, I'm going to put the link for you to watch down in the description box. So all you have to do is click on that description box, watch that video. So right now, what I want you to do is pause this video, look at the description box, click on that video, watch the animation, and then come back here and we will continue talking about lens accommodation. All right, so I hope you liked that video. Now, when we're talking about lens accommodating, we're talking about the lens changing shape. So the lens can thin or bulge, and the ultimate goal is to get the light rays to converge on the retina. Now, we're first going to talk about thinning. So when the lens thins out, this image right here, when it thins out, it will thin out when we are trying to focus, getting light rays to converge on the retina, when we're trying to focus on things that are far away from us. Okay, so far away. So take a minute, look at something pretty far away. And what's going to happen is your lens is going to thin out. Now, when it thins, remember the light rays are going through. When the lens thins, it's going to decrease the refraction. It won't bend as much. So we say it decreases the refraction of the light. Now, if we're going to read something, right? If I grab a book and we're going to read my binder, and I'm going to start reading from a book or something up close, then my lens has to accommodate at that point as well. My lens will bulge. It would become thicker. And this happens when we try to view things up close. Now, when it becomes thicker, it's going to increase the refraction. It will increase the amount of bending. Remember, the goal is to get those light rays to converge on the retina so that we can have the best vision. If, for some reason, it does not, if the lens is trying its hardest to thin out and to bulge, but it just can't get there, those light rays aren't going to converge on the retina. And whatever we're looking at will not be in focus. Okay? So theoretically, if we keep tracing out those light rays, depending on which if it's not thinning or it's not bulging, it's theoretically going to be converging somewhere beyond the retina or before the retina. Either way, it's not going to be in focus for us. Okay, so we want our lenses to be very pliable. Okay, we want them to be very pliable. All right, so the next thing we're going to talk about are some refraction and accommodation disorders that can occur in eyes. And the first one is myopia. So that first word down there, myopia. Now, probably most of you have heard of these, but maybe you just didn't know the technical term for it. So first of all, myopia. Myopia is also called nearsightedness. So nearsightedness. This is when we can focus up close, but we can't really see things in focus that are far away. So we're nearsighted. It tells you how well you're getting it into focus. We're nearsighted. Okay, we can't see things far away. They're blurry. Things are blurry far away. That's technically called myopia. And that can happen because of if the lens doesn't properly bend, it doesn't thin out or it doesn't bend. It doesn't properly thin out, then that can happen. The next one is hyperopia. So hyperopia is farsightedness. So we can see far away in focus, but when we go to read up close, when our lens needs to bulge to increase that refraction, it just doesn't work well enough. And so theoretically, those light rays, if we were tracing those, they're going to be converging somewhere beyond the retina. So even though we're going to try to read here and try as much as we can to get that lens to bulge, therefore refract the light, still not going to be in focus. All right, the next one is presbyopia. So presbyopia, and presbyopia is a combination of both. It's actually a combination of both myopia and hyperopia. So it's a loss of lens accommodation. For whatever reason, your lens just will not accommodate as it once did. Now it could be you could be born that way, right, from the beginning, or a lot of times this will increase with age. So the lens just it isn't as pliable anymore, and it's not going to thin out as much as it did, and it's not going to bulge as much as it did. So guess what? It's really hard to see things far away, and it's hard to see things up close. All right, so you have that's kind of the best of both worlds, or the worst of both worlds, presbyopia. The next one is called astigmatism. Now, this word, I hear this word pronounced incorrectly a lot, and the word starts with an A. So the word itself is astigmatism. Okay, it's not a space stigmatism. The whole word is astigmatism. And this occurs because of unequal curvatures of the cornea. Now, technically, if you're looking in a textbook and you look up astigmatism, it just means unequal curvatures of anything that the light has to go through. And I told you the light had to go through two major structures. It has to go through the cornea and also the lens. So, but 90% of the time when we have an astigmatism, it is the cornea. All right, so the cornea is not quite symmetrical. So one little part of that cornea is just not as symmetrical as the other side. And we start to see maybe a little patch in our vision that is blurry. Okay, so that's what we have when we have an astigmatism. Now, these can be corrected with corrective lenses. All right, so probably some of you have maybe myopia or hyperopia or maybe even astigmatism, and those are usually corrected with some corrective lenses. Now we're going to talk a little bit about the photopigments. Now I want to take you into the retina. Into the retina, we have the photoreceptors. Inside the photoreceptors, we find photopigments. Photo just means light. So with light as the stimulus for vision, remember I said that there's lots of different stimuli that will activate receptors. In this case, it is light that is going to activate a receptor. So how in the world is it going to activate that receptor? The receptor is going to use pigment in order to absorb the light. So that's what we're talking about, these photopigments. The photopigments are located in the rods and cones, okay, and we're right up here. These are just a little depiction of rods and cones. And this is the first step in transduction of the stimulus. We have to absorb the light. So the photopigments are used to help absorb the light. Now, the photopigments, there's two major parts to the photopigment. There is an opsin, a molecule known as an opsin, and there's various types of opsins. And then there's another molecule that's called retinol with an A. So retinol with an A. And retinol is a vitamin A derivative. So you may have heard of that before. Okay. So we have two different types of photoreceptors. Each type has opsin and retinol as their photopigments. They may have slightly different opsins. So that's what we're going to talk about now. So rods have their photopigment is known as rhodopsin. That's kind of easy to remember. Rods, rhodopsin. And remember what I said in the other lecture is that rods are activated in dim light. Okay, they are highly sensitive to light. So you just need a tiny bit of light in order to activate our rods. Now, once the light, we're staying with the rods just for a minute. Once the light hits the rod, for example, that light has to be absorbed, and it's absorbed by the rhodopsin, okay, right? Because I said the rhodopsin is the photopigment, and the light has to be absorbed, and this is how it's done inside of this cell. Okay, now on to cones. Cones is the other type of photoreceptor. Cones, we have basically three different types of cones. We have, or there's three varieties of cones, and it's blue, red, and green. Okay, that's why I have the B, R, and G up there. So blue, red, and green. Now the pigment inside of cones, it's slightly different. It has a different type of opsin, so we give it a different name, and it's called photopsin. You would think it was called conopsin, but they didn't ask me. But it's called photopsin. So cones, the photopigment inside the cones are called photopsin. There are 3 varieties, and it's blue, green, and red. All right, so this picture up here is, it kind of shows you what these photoreceptors look like. This is a really old drawing way back when in textbooks. We just had like pencil drawings. That probably predates me, but they just had pencil drawings. So a lot of times we just had pencil drawings of these images and of these, minute body structures. But I just thought I'd throw that one up there just to show you, just even in a pencil drawing, it can show you a lot. Okay, now we're going to start getting into the nuts and bolts of how vision occurs. Okay, how do we activate these receptors? All right, we've gotten the light there to the retina where the photoreceptors are. Okay, now what happens? Well, remember that process that we talked about where the receptive field must be stimulated. Okay, that's been done. What was the next part of that after the receptive field must be stimulated? Then we have to have transduction of the stimulus. Transduction of the stimulus means we have to have some sort of conversion, right? Because light rays aren't going to get to our brain, but nerve impulses are. So transduction of the stimulus, you're going to do a conversion of the energy and light, in this case, into a local potential. All right, so now we're going to start talking about how does that happen. And as we go through these processes, we're going to do it with the rods as our example. The cones use a slightly different photopigment. However, they function the same. All right. So I'm just going to use rods as our example, and you're going to see that in the pictures. So we're starting out with light, and light has gotten to the photoreceptors. Now that light has to excite the photoreceptors. We have to absorb the light. They have to absorb the light. And we call this particular process excitation of the photoreceptors. We're activating the photoreceptors, basically. Another name for that is called photoreception. All right, so before we jump into this, I need to backtrack a little bit and talk about those photopigments. So what was the name of the photopigment in rods? Because we're using that one as an example. Rhodopsin, right? Starts with an O, an R. It was easy to remember. Okay. And it's made up of opsins, different types of opsins, and retinol. Okay, so I'm going to attempt to draw okay, when we're looking at this molecule, because we're going to be talking about what does the photopigment do, and there's 2 parts of it. So I'm going to draw a circle, sort of. That's going to be my opsin. That makes sense. O for opsin. Then we have a retinol. And so the retinol, think of a lowercase r. Okay, that's how I remember it. I'm going to draw a lowercase r. Okay, that's the best I got for drawing on the computer. All right, so inside of a rod, we have that photopigment, rhodopsin, made up of opsin and retinol. And they are attached just like this. Now, the process by which we activate the receptors, and we use the photopigments in order to do that, to absorb the light, it is a cyclical process. So that's why you see the circle up here. It's a cyclical process. So I have to jump us in at some point during this process, and where I'm going to jump in is right in the dark, right before the light comes on. Okay? So if we were to take a look inside the rod at the pigment, right before it is exposed to light, it's going to look like this drawing. Okay, the pigment will look like this. It's made up of opsin, and what was the other one? Retinol. Okay. Now, there in the dark, the retinol part it forms a bent part of a molecule, if that makes any sense. The molecule actually is bent. That's why I drew it like this. And where I am is right up here. So you see me circling up here, and you see something that kind of looks like my picture. That's where we are. This pigment, when it looks like this, we have a name for that, and we call it cis-retinol. So right up here, cis-retinol. Let me try to pick up no? All right, we're just going to go with it. And there's the name right there, cis-retinol. All right, so now we're flipping the lights on. We're flipping the lights on, and what happens? Now there's light, it's hitting the retina, it's getting to the photoreceptors, it's inside the photoreceptor sort of, and it's activating the photoreceptor. It's going to be absorbed by the photopigment. Now, it's one thing to say it's absorbed by the photopigment, but we really have to know chemically what's going on. So this is the pigment, and when we flip the lights on, see if this is going to work, what happens is that the molecule changes shape. So it goes from the cis-retinol shape to a trans-retinol shape. So a trans-retinol shape looks like this. Okay, it just straightens out. And that's what we're seeing right over here. We call this one cis when it's bent, and this one trans when it straightens out. So what has happened when the photoreceptors, the photopigments, start to absorb the light, it undergoes a cis to trans conversion. All right? That's the first thing that happens. So the light is physically changing that molecule. We get a cis, that one, to trans, that one, conversion. And we have a name for that. That is called isomerization. So isomerization is just when the cis-retinol gets transformed into trans-retinol, gets changed. It basically straightens out. And this is where we are here, okay? We're still in the light. So as the light stays on, and we'll see here, what happens is it's been absorbed. Now the light has been absorbed. Molecularly speaking, what has happened is the light has triggered a change in the shape of a molecule, the pigment. Now what happens is that the trans-retinol will completely disassociate from the opsin. So it's really bad. They come apart. Okay, they break apart. And where are we seeing that? We're seeing that right here. Okay, so they're breaking apart. Now, when trans-retinol completely separates from the opsin, that's what I drew here. Now, what happens is that, remember I said it was cyclical. Now an enzyme comes in and will reattach. That enzyme will rebend this molecule. Okay, it rebends it to cis and then attaches it. So if I were to draw that, here comes the enzyme. It bends it and it reattaches. Kind of looks like a cherry there. But so it reattaches. Now we're kind of back over here and it has regenerated. So when it straightened out and when it straightened out, it had absorbed the light. When it broke apart, it separated from those two parts of the molecule separated. It had to be put back together to its original. An enzyme helps it do that, and once it's put back to the original, it's now able to absorb more light if there's more light coming in. Okay? So it's basically, what is that light triggering the molecule in the photoreceptor, the photopigment, to do? That light is triggering a change in the shape and formation of that molecule. And that is our absorption of the light. Now, this is not all that occurs. This is just the first part, because we have to absorb the light. And if we think back to that process we talked about, the receptive field must be stimulated, got it. We have to have transduction of the stimulus, okay? That means we have to convert the energy in light into a local potential. And we're not there yet, right? We're just kind of in the middle of that transduction, okay? All right, so that's what we know so far. Next slide, we're going to talk about what else is going on inside of that photoreceptor. What else is going on inside of that photoreceptor so that we can understand what happens next. All right, so I've been teaching this a while, and what I've found out is that there's a better way to teach this material, and hopefully to get you to understand what happens next. So we're talking about light absorption in the photoreceptor, and you just learned about the pigments and how and what role that they play in that light absorption. So in order to find out what happens next, the best way I've found is to kind of take a step back and teach you what happens in the photoreceptor in the absence of light, okay? Because to truly understand what happens next, you have to understand first what's going on in the photoreceptor when there is no light at all, in the absence of light, okay? So in the dark. So where I want to take you to is the plasma membrane of the photoreceptor. And for our example, we're using a rod as the example, but this works for cones as well. And so the pictures that you see up on the PowerPoint is a picture of a rod. But where I'm going to take you to the plasma membrane of that photoreceptor, okay, in the absence of light. And you're going to learn about what's going on there at the plasma membrane. So in the dark, the first sentence I have up there is sodium channels held open by cyclic GMP. There's a new molecule I want to introduce you to, and that is cyclic GMP. GMP. You don't have to know what it stands for at this point. Now, the first sentence read, sodium channels held open by cyclic GMP. We are at the plasma membrane, and you know that there are sodium channels in the plasma membrane. So I want you to visualize that sodium channel, and right at the top here on the left is showing you a sodium channel. Okay, this little pink one, right? This integral protein here in the phospholipid bilayer. So that is a sodium channel. You guys know how sodium channels work. And that sodium channel is being held open by a molecule known as cyclic GMP. So in the dark, this is what's going on. All of the sodium channels are being held open by cyclic GMP. So if a sodium channel is being held open, that means sodium is allowed to enter into the cell. Okay? So sodium enters the cell. If cyclic GMP is holding it open, sodium is rushing into the cell. Now, so sodium's rushing into the cell, and as it turns out, potassium goes out. So sodium's going in, potassium's going out. You've heard this before, right? When sodium goes in and potassium goes out, and you learned that as part of a depolarization event. So that is what's happening. You're getting a depolarization of the photoreceptor, the rod or the cone. So you might think, well, wait a minute, New. You just told me this is the absence of light, and we're in the dark. Why would the plasma membrane be depolarizing? Because if the stimulus isn't there, why would it be depolarizing? I didn't mess this part up. This is actually what happens. It's just that it's counterintuitive. So, as it stands, in the dark, the photoreceptors are depolarizing. Okay, so in the absence of the stimulus for vision, the photoreceptors are depolarizing. Now, we have the photoreceptors, and the photoreceptors are going to form a synapse with another type of neuron. And that neuron is known as a bipolar cell. So let's back it up again. Take a look at the picture. We have a rod here. It's being held open. The sodium channel is being held open by cyclic GMP. Causes sodium to go in, potassium to go out, and you're getting a depolarization event. Now, as a result of this depolarization event, the rods and cones, the photoreceptors, are going to release a neurotransmitter. Okay? So they're going to release a neurotransmitter known as glutamate. That might be the first time you hear that word, glutamate. So glutamate works in the synapse between the photoreceptor and the bipolar cell. So we have glutamate there in the synaptic cleft. Glutamate gets picked up by the bipolar cell. So now I'm here at the bottom sentence. The glutamate is actually inhibitory. Remember, you learned about excitatory and inhibitory. In this case, the neurotransmitter is inhibitory to the next cell in line, which happens to be the bipolar cells. So since glutamate is inhibitory in this instance, it's not going to stimulate the bipolar cells. So if you don't stimulate the bipolar cells, you have no receptor potential. If you have no receptor potential, you have no action potential. If you don't have an action potential, you don't have a nerve impulse. Now, that part probably makes sense, right? So, like I said, in the dark, in the absence of light, the photoreceptors are depolarizing, okay? And it has to do with that cyclic GMP. So they're depolarizing, as a result, releasing that neurotransmitter glutamate. Glutamate is inhibitory, and so it causes an inhibitory postsynaptic potential in the bipolar cell. Since it's inhibitory, there won't be any receptor potential. So no receptor potential, therefore no action potential. No nerve impulse to the brain. Okay, that makes sense. So that we need to know before we go on to the next part. All right? Because the next part is kind of picking up where we left off from the last slide, is what happens next. The light, going back to our process, the stimulus has activated the receptors. Okay, receptive field must be stimulated. We've done that. Transduction must occur. We've only done the first part of transduction. And the first part of transduction in a photoreceptor is the absorption of light. So you learned about that with the photopigments. So the photopigments changed shape. Remember, they broke apart. They were reformed. It was a cyclic process. And so that's not all that has to happen. That's only the first part. So at this point in the rod, because we're using that as an example, what happens is that we have absorbed the light. It has absorbed the light using the pigment. And at that point, it's going to kick off a series of events, like a domino effect inside that photoreceptor. So the light activating the receptor, being absorbed by the photopigment, now it kicks off a series of events. So now we get to learn what happens next in the light. Okay, now we're back in the light. The light came in. It was absorbed by the photopigments. It broke apart. And now what happens? Now we're right up here at the top. Now we're in the light. Now what happens? So when I say it kicks off a series of events, think of it like a cascade event, or visualize the domino effect, right? One thing has to happen, then another thing has to happen. That's what it is. So the light was absorbed with the photopigments, and as a result of that, an enzyme comes in and breaks down the cyclic GMP. All right. All right, now we're in the light and we're finding out what happens next. In the light, after absorption, the enzyme is going to break down the cyclic GMP. Remember what you just learned about cyclic GMP? It was holding open the sodium channels. So if cyclic GMP is being broken down, it's no longer going to hold open the sodium ion channels. So no more sodium rushing into the cell. However, potassium is still going out of the cell. So no sodium going in, potassium is rushing out. Now, if we were to go in and really take a look at the charge, because of that, okay, potassium, what happens is if the potassium is still going out, then that's going to result in a hyperpolarization of the rods and the cones. So instead of depolarization, it results in hyperpolarization. Remember, hyperpolarization, that should remind you of the action potential phase, the phases of the action potential. So when a plasma membrane is hyperpolarized, that means it's even more negative than resting membrane potential. So backing up a little bit again, the cyclic GMP is broken down, and this is in the presence of light. No more sodium rushing into the cell. Potassium is still going out. So because of that, it changes the charge. It's going to make it even more negative. So that results in a hyperpolarization event inside the photoreceptor, the rods or the cones. Now, the photoreceptor will synapse with the bipolar cell. Now, if the rod or cone is hyperpolarized, it's not going to release a neurotransmitter, glutamate. So it's not going to release the neurotransmitter glutamate. That makes sense. So the lack of glutamate in that synapse does something though. And what it does is it excites the bipolar cell. Here's where it gets a little weird. So the lack of the glutamate in the synapse is going to kind of activate the bipolar cells. It excites the bipolar cells, and it causes the bipolar cell to have a receptor potential. Okay? So now we have a receptor potential in the bipolar cell. Now, let's back up again to the process. So back up to the process where we have the receptive field must be stimulated. We got it. Transduction of the stimulus. Let's think about what the transduction of the stimulus was. It is where the energy of the stimulus gets converted or transduced into the energy of a local potential. So are we there yet? We are there because the bipolar cell, the lack of the glutamate in the bipolar cell, what it did was excited the bipolar cell. So the lack of glutamate at that synapse excited the bipolar cell. Okay, and as it turns out, the bipolar cell will now experience a receptor potential. A receptor potential is a type of local or graded potential. So now we have our graded potential. We've gone through the second one, the transduction of the stimulus. What's next in that process? Okay, we have to stimulate an action potential. So the bipolar cell synapses with a cell called the ganglion cell. So now we are right down here. The bipolar cell synapses with a ganglion cell. And it is the ganglion cell that can initiate an action potential. So it forms a synapse if we're exciting the bipolar cell into that bipolar cell having a receptor potential. It is going to, in turn, excite the ganglion cell to depolarize and therefore have an action potential. An action potential in a ganglion cell is our nerve impulse. Once that triggers the action potential in the ganglion cell, our nerve impulse is now sent to the brain. Okay, so now we have a nerve impulse being sent to the brain. That makes sense because we're in the light, right? Because the photoreceptor has absorbed the light, kicked off the series of events to a point where we have transduced, and then we have activated the ganglion cell. We have triggered an action potential in the ganglion cell, which is the nerve impulse, which is being sent to the brain now. All right, so now we are picking up where we left off, and we're going to talk about the pathway to the brain. So at this point, you have an action potential that is being sent along a ganglion cell. And axons of the ganglion cell basically all meet up, and they all exit out the eyeball at the same place. They meet up, exit out the eyeball, and what they do is they form the optic nerve. So you see where I'm circling right here, there's an optic nerve here, an optic nerve here. Now I like this picture because it has some colors on it. Now we know that the ganglion cell is, the axon of the ganglion cell is transmitting that nerve impulse, and it will take it to the brain. So here we are at the optic nerve. Now notice this one is blue. Now what I do want you to notice is that some of the impulses will stay on the same side as the brain, but some will actually cross over. And where it crosses over is there at what's called the optic chiasma. So if we look at the other side, which is yellow, some are crossing over there at the optic chiasma. Some are staying on that path. Now, the optic chiasma over here on this dissected brain, you'll be able to see it here. It looks like a little X. That's because the optic nerves have been cut. So you're just seeing it as that little X. So if I were to ask you what's going on at the optic chiasma, you can tell me that some crossing over occurs of the axons of the ganglion cells or the nerve impulses. The nerve impulses that are coming from that eyeball are crossing over to the other side of the brain. Now, once we pass the optic chiasma, we no longer call it the optic nerve. It's actually called something else, and it's called the optic tract. So now those nerve impulses are traveling along the optic tract, here and here. Now, it is at the optic tract where diverging starts to occur. And the best way I can explain what diverging is kind of like some of the nerve impulses are getting off the highway and going into a little town. Okay, so they're dead ending in a different part of the brain. And you see that here. All right, so you see some of the yellow is kind of going here to a midbrain area. Some of the blue is going to the midbrain area. So diverging starts to occur, and some visual processing occurs there in the midbrain. Now, the majority of the nerve impulses basically stay on track. You can say, I shouldn't say that. They basically, they don't diverge. That's better. They don't diverge, but we now give it another name. And so we call it the optic radiation. So where I'm circling here would be the optic radiation. Over here on the dissection, here's the optic radiation. Just one side, the other side's been cut. So now the impulses are traveling along the optic radiation, and they will travel along that optic radiation until they hit the primary visual cortex, or synapse with another type of cell at the primary visual cortex. So the primary visual cortex, that is located in the occipital lobe. You remember the occipital lobe, right? Right back here. That's where we find the occipital lobe. So the majority of the processing for your vision occurs in the primary visual cortex of the occipital lobe. That sounds like a test question. So where does the majority of the processing for your vision occur? That's right, the primary visual cortex of the occipital lobe. All right, so I think that's it. Our last slide, let's see if I can get this to advance. The last slide has a bunch of questions on it, and I thought that you could use those to kind of help you study. These are good questions to practice with. And I'm pretty sure all of them you can find in these 2 lectures on the eye and vision. Let me just look over them real quick. All right, so I think that all of those questions can be answered by listening to and taking notes on the 2 lectures, part 1 and 2 of the eye and vision. Now, if you do have any other questions, write them down, bring them to class. We'll spend a little bit of class time talking about it, and hopefully I can answer all of your questions. Don't forget, if something that I talked about in these lectures don't really make sense, you have your textbook as well, right? So this material is covered in your textbook, and it's a good idea to read over those pages that cover that same material. All right, so I will see you in class.